Hi, we're going to go ahead and start experiment number 3 now. And in experiment number 3, we are looking to see the signs of starch production, which has occurred in some of our leaves as a result of photosynthesis producing sugars and a need to store those excess sugars. I'm going to begin here by taking a leaf off of a plant. And so this plant has been recently exposed to light, but we did keep it in the dark for a while so that it would stop photosynthesis. Then we began allowing it to undergo photosynthesis again with a shield on the leaf to block the light from certain locations. So I'm going to take that over here next to another leaf that I had. And I've got a couple of options, but I need to know where the position is of my particular leaf shield. So I'm going to go ahead and take a photo of this just to make sure I know where those leaf shields were. Of course, if I don't have a camera to take a photo with, I can go ahead and draw the positioning of my leaf shield. And once I'm done with that, I'm ready to go ahead and take those shields off. And I can place my leaf in boiling water for 5 minutes. Now, the important thing that I'm going to do here is I need to break these cells open, because ultimately, if I'm going to see changes in this leaf, I need to make sure that I'm releasing the chlorophyll so that I can see any underlying changes that are going to be there. So this will go for 5 minutes. Then I'm going to move them over to this station where I'm going to boil them in alcohol for 5 minutes. This should remove all of the coloration in my leaf, giving me essentially a white leaf to take a look at. The reason I want to do that is that I can then move over to my third station, and here we have some iodine which I'm going to place in this dish. I can soak my leaves in that. And if we think back to an experiment we did earlier in the semester, we know that we're going to have a reaction with starch and iodine where we should get a black or a kind of black-blue coloration indicating the presence of starch. So that hopefully will allow me to figure out which parts of the leaf have been undergoing photosynthesis and which parts haven't. Okay, so it's time for me to take my leaves from the water and move them over into alcohol. I'm just going to carry them over here next to it. I can grab my leaves one at a time and place them into this alcohol. Now the alcohol has been set up in a double boiler, so we have water boiling on this hot plate, and we have a beaker of alcohol, which we can see here immersed inside of that. Obviously, alcohol is going to boil at a lower temperature, and we want to make sure we avoid any fires. So we'll take this out of here in 5 minutes and transfer it to the iodine. Okay, so it looks as if it's about time to move my leaves from the alcohol over to the iodine. We should come take a close look here at the alcohol, and you'll notice that the alcohol has dramatically changed color as it has extracted the pigments from the leaves. I'm going to go ahead and pull out my first leaf here, and you can see that this thing is nice and white. We've extracted the pigment from it, so I'm going to go ahead and lay that over there in the iodine. I'm going to grab my second leaf. I've just done 2 leaves to make sure we get a nice result. We can take a look at both of them and see which one comes out better. So you just need to track one of these from your initial photograph or drawing till the end of this. I'm going to go ahead and put those in the iodine and make sure they are fully immersed. You can already start to see some of these areas darkening as we have a reaction between the starch and the iodine.